Now it's time for a presentation of Real Research. Real Research is a small but rapidly growing biotech startup founded in 2017 by two alumni of one of the best universities in Poland, Jagiellonian University. We are determined, they say, to change the way people carry out research, moving it away from inefficient 2D cell cultures to the much more reliable 3D cultures. Ladies and gentlemen, let's now meet Real Research.
Hello, I'm Marcin Krzykowski. I'm the CEO of Real Research, and the mission of our company is to revolutionize life science. This is the problem that we are facing today in the drug development business, basically. There is ever increasing rising costs of new drugs production, new drugs creation, because of extremely low success probability and extremely high investment. Back in 1970, you used to have to spend 150 million US dollars to really create a new drug. When we started, it was about 2.2 billion, and now it's 2.6 billion dollars to create one new drug. So how does it look? If you really want to start a pharma company, you will start probably with about like 10,000 different drug candidates. And then through all the preclinical process and drug screening, you will narrow it down to 250 and then to 5. And at the end, maybe you will end up with one working drug. It will take you up to 20 years. It will cost much more than $2.2 billion. And the chances of really finding the right drug is much less than 5%. And this is a problem which is unacceptable. So what do we offer to solve the problem? Ligel is a technology platform that allows us to create customized hydrogels for 3D cell cultures. But what does it really mean? If you look at the first part of the drug development, I mean the first one where you have all of those 10,000 drugs and you narrow it down to those 250 and then to 5, the first elimination, that's where you actually remove from your pool of possible drugs 97% of the possible candidates. And there is a huge problem in that stage of drug development, because even though you remove 97% of the possible candidates, most of the times companies are using very simple, flat, two-dimensional cell cultures, as you can see on the presentation. On the left-hand side, you can see normal cells growing on flat plastic. It has been done like that for the last 50 years. And to be honest, it gave us some interesting results, but it's not working anymore. The time has changed, and we should change the way we are doing our first step of drug development and move to three-dimensional cell cultures, which you can see on the right-hand side. Let me show you a case study where you can clearly see a difference between a drug that is working or is not working. And as an example, I will use PANC1 cell line and 5FU as the tested drug. Again, on the left-hand side, there is a 2D cell culture where in the first column you see day 0, there was some amount of cells. In 3 days, these cells grew to like more than tripled. And in the same time, when you added the drug, you see about 50% inhibition of growth. That would be great. But if you look at the same drug, the same number of cells, the same number of cell divisions in 3D cell culture, you can actually see that, again, on day 0, you have certain number of cells. Day 3, without the drug, obviously there is nice growth of cells, but day 3 with the drug, there is basically no inhibition. Like there is just a little bit, but nothing really special. And what's so amazing about this result is that 5FU should not work in this setup. This is a pancreatic cancer where 5FU is not used. So it means that if you would be researching a new drug, in this exact case, you should actually exclude the drug from your pool, not include it in later research. And as you can see, the 3D cell culture is the way to get that knowledge exactly from this example. So why do we observe those differences? So 2D cell culture is a flat plastic. It's extremely stiff. There is no basically very little cell-to-cell communication. And like, it's actually very well known that about 95% of the positive results in 2D are not working if you go down the line. Comparing that to 3D cell culture, in 3D you can mimic the natural environment of those cells. You can enhance cell-to-cell interaction and therefore change the response to drugs. You can reduce also the number of animals necessary for your research. And there is a really wide range of application of those cell cultures. So just to show you, for me, one of the most amazing publications there is in our topic. On the left-hand side, you can see a result from a publication comparing a full gene expression profile of about 30,000 genes, comparing 2D with in vivo and 3D. So the red column is actually, the red color is actually giving you information about the downregulated genes. And as you can see, the red column is actually the two-dimensional cell culture, whereas on the left-hand side of the graph, you can see 3 green columns. All of those 3 green columns are consecutively 3D cell culture after 11 days, 3D cell culture after 7 days, and in vivo. So they are not exactly the same. But if your drug is targeting a certain gene or certain protein, you have actually much greater chance of really having that gene being expressed, comparing to two-dimensional cell culture, where you can see that 80% of those genes are heavily downregulated, what will influence the working, like the function of the drug and the target that you're aiming at. And obviously, this here you can see again that on the right-hand side that the 3D cell culture showed much lower response to for example, oxaliplatin in this case. So let's go on. Obviously, we are not the first ones to really introduce 3D cell cultures, but to be honest, we are the best ones. Let's look at our competitors. Competitor A is like one of the most common competitors that's making the hydrogels from tumor-derived materials. And it was a great tool for the last 20 years, especially for research purposes. But it's not fulfilling the expectations anymore, because it's actually not really repeatable, because every batch is different. It's contaminated with growth factors, and it's very hard to scale this product to large-scale testing. If you look at a competitor B, it's actually one of the most common types of plastic that is actually a little bit shifted just to allow for 3D cell culture. So you don't really get all of those possible benefits of 3D in long term. And our competitor C has a great advantage of being nicely repeatable, and it's also nicely transparent, for example, like the competitor A. But the biggest problem with competitor C, it's actually not protein-based. It's based on plant-derived material, which is, again, far away from what we are. We are not derived from plants. We are made of proteins, and that's why the hydrogels that we use for testing should be made from proteins as well. So what do we do as LifeGel? So first of all, just to refer to what I said about our competitors, we are highly reproducible, basically batch-to-batch reproducible. There is no contamination in our hydrogels, and we can easily scale the manufacturing of our hydrogels to basically almost any demand. But what's really the most important part of our product is that it can be customized to almost any application, which means that you don't get just one product for everything. You can have a product a little bit different, a little bit fitted to research on pancreatic cancer, on brain tumors, on bone tumors. And that's actually the biggest benefit, because multiple publications have shown already that you need to have that the way you culture cells, if you recreate the natural environment of those cells, you can actually get much different result between different types of tissue. And that's the key in terms of doing the early stage drug screening. So let me now jump into what do we actually offer to our clients. We really concentrate only on the very, very first stages of clinical research, of preclinical research, sorry. So in the part where you actually looking for the target to hit or hit to lead, it means that's the moment where you invest roughly about $100 million to test all of those, let's say 10,000 potential drugs. And then you invest about 5% of the entire amount, and based on those 5%, you will decide whether to invest another more than $2 billion. And here's how we can actually help our clients. First of all, if you add to the first 100 million, if you add $100K, you can actually increase the probability of finding the right drug, simply because the sieve you will use to select those candidates will be much thinner, and therefore much fewer drugs will go to the third and fourth part of the column. What's also important, you will immediately save a lot of money, simply because in the second phase, you're going to spend about 200 million. And if you imagine that normally you would get 250 potential candidates, and now rather than 250, you will actually end up with 100. You will immediately save like 60% of the 100 and of about 200 million. So the investment will return immediately, no matter what the real result is. And to be honest, if after doing the high throughput screening at the very beginning in more advanced 3D cell cultures, if nothing will come out of it, this is actually the best result, because then you're not going to invest, and you will be able to spend this same funding for another more useful, trials that you're actually working on. And at the end, if you will finally find a drug and screen it additionally in 3D, and the same drug will go through all the investment, which should be already much lower, at the very end, you increase the probability of really finding the drug much more above the 5% that's usually on the market. So just to sum it up a little bit, by the initial investment of about 100K to the 100 million that you already spent, you can increase the chances of success by at least 50%, simply because you just narrow it down, and those not working drugs will be eliminated very early. You will also lower the cost of investment for future preclinical studies and later on as well, again, simply because there will be much fewer drugs to work on. And just to be fair, if none of them will work, it's good to kill the project early. So our business model is basically that we work with academia and businesses. We do have small academic clients, small EU pharma and medium EU pharma, also CROs and startups. But also as a B2B, we work with big pharma and much bigger CROs. This is our team. I'm Marcin Krzykowski. I'm the CEO of Real Research, but we have also many experienced employees and founders and co-founders who are working hard every day just to make our mission come true. And we want to truly revolutionize life science by introducing the 3D cell culture in much larger scale than it is today, simply because our product is the first one that can be actually highly reproducible and used in a large scale. This is a short list of our clients, co-workers, cooperators, and the list is actually much bigger. And for example, we are also working on one COVID application where we want to increase the true positive readout of the diagnosis. So there is a lot of going on outside of the core business, and I'm excited to learn about what you're doing and what you're interested in. And thank you very much for your attention, and feel free to contact me and here you can see my data. Thank you very much.
As with all research, using cell culture technologies in a relevant way is always a challenge. The success of drug discovery and drug development is highly dependent on how experiments are planned and executed. In the human body, cells form three-dimensional or 3D cell structures. 3D cell culture allows researchers to mimic these physiological conditions in vitro. Science is shifting into 3D cell culture for drug treatment as well.
At Real Research, we believe that the real breakthrough in life science is yet to come. Today's reality is that despite ever-growing spendings on drug development, every year fewer and fewer drugs are being developed. This stagnation has to be overcome. We envision a different future. We see a future where a well-designed in vitro model correctly mimics the disease, and later, when the drug works in vitro, it will work in vivo and in clinical trials. Success of our clients is also our success, and eventually, as people, we will benefit from new and better drugs. Our approach is simple. We explore the problem set up by our customer. It can be anything, from setting up a basic disease model to much more complicated maximization of a gene expression based on 3D environment. We can do small screens in our lab, but we can also outsource the protocol to our client's lab, as well as to a CRO.
Real Research is located in Krakow, in one of the biggest life science centres in Poland. It's a startup biotechnology company initiated in 2017 by two enthusiastic founders. A mission of Real Research is to revolutionise the life sciences. In the last 3 years, Real Research has managed to build an international team and is still growing. Revolutionary products have been developed based on innovative inventions and a successfully filed patent application. Real Research believes there's a great future ahead of them. Real Research provides customers with ready-to-use hydrogels for 3D cell culture called LifeGel in a range of multi-well plate formats. In this LifeGel environment, cancer cells, for example, spontaneously aggregate and form spheroids. We recognise the importance of our clients' time. This is why we work for you to help you achieve reliable and meaningful results. Work smart, not just hard.
Hello, my name is Dave. I'm a scientist. I work in the Real Research Lab. Have you ever thought why drugs are working perfectly in 2D cell culture in vitro, but fail the in vivo study? More than 95% of promising drugs fail in animal studies and clinical trials. Have you ever considered testing your drug in 3D cell culture instead of 2D? Why should you? Cell culture is a primary tool in drug discovery. Currently, a research paradigm is shifting from 2D to 3D for cell culture. Why? Imagine that a steamroller drives over me. At that stage, I'm completely flat lying on the surface. But 3D is our natural state. In the human body, cancer cells grow in three-dimensional structures. At Real Research, we believe 3D cell culture is the future of life science. Let me introduce you to LifeGel, a first of its kind, highly repeatable protein-based hydrogel for 3D cell culture. We are made of proteins, and research should also be in a protein environment. Our bodies are made of various niches. Every organ creates a different environment for cells to live. With LifeGel, we can mimic those environments to let cells feel at home. In short, LifeGel can be customized to mimic multiple niches to find the best fitting hydrogel for your research. Cancer cells growing in their original microenvironment will exhibit their natural traits and therefore respond to drugs as if it were happening in the organism. The same microenvironment can be used to culture healthy cells, for example, stem cells. As a client, you get ready to use hydrogel on a 48, 96 or 384 well plate. Plates are ready to use by the scientist with RTU LifeGel. LifeGel is a protein-based hydrogel. It is batch-to-batch repeatable, free from any growth factors, and compatible with standard imaging platforms. You can read your assay using a standard microplate reader, or a more sophisticated imager if you want. For example, you can do cell titer blue, cell titer glow, or Hertz measurements. LifeGel is highly transparent and therefore perfect for cell imaging in many different scenarios. You can use it for drug discovery, drug development, drug toxicity, and even bioproduction. You can culture multiple cell types, and you can do all of it using automated lab equipment like an OT2. For example, when you do IC50s in 2D cell culture, you will get a very nice curve. Unfortunately, often clinical data shows that patients need much higher doses because in 2D, each cell immediately gets 100% of the drug. In 3D cell culture, we can mimic how it is really happening in the human body, and therefore, IC50s from 3D cell culture will have much greater predictive value. LifeGel is a tool for your effective preclinical drug evaluation. With LifeGel, the next time your in vitro results look promising, you can be more confident in great in vivo results and in clinical trials. At Real Research, we love what we do and we are excited about the future. LifeGel, revolutionizing cell culture. www.real-research.com